gene product around the cell. That's more difficult, but it can be done by this HER process. So the cells do this for you. All you have to do is make the break in DNA. And this was the fundamental kind of sticking point in this whole technology. The idea was there. How do you make and program a nuclease to specifically recognize gene A and not all the other tens of thousands of genes that are in the organism? So you have this kind of dream enzyme that you want to be able to produce. It has to be specific, site-specific DNA recognition of a single site within the genome, so not bind anywhere else. So you don't want to get any off-target cleavage effects. Remember I told you that double-strand breaks are toxic? What you don't want is a protein that just chops your genome up into lots of little pieces. You want a break at one location, the location you're interested in, and that's it. You want that break to be introduced at that site, and that sounds a bit sort of trivial, but what you don't want to have is a recognition at one site and then a cleavage that somehow gets located somewhere else on the DNA. It has to happen where you're recognizing, because you want it to recognize gene A and cut gene A, not have some kind of bystander activity which cuts other bits of DNA. It's got to be relatively simple, so ideally a single gene. So if you can make a single protein that could do this, that's a lot easier to deliver than saying I've come up with a system that requires 100 genes. That's going to be really impossible to get into a cell. So you have a simple system, one gene will do the job. And you want that gene to be expressible in multiple different hosts. So ideally, if you wanted to say, well, I've come up with a new system and it works in human cells, it works in coli, it works in plants, it works in yeast, fantastic. That means more people can use your technology. And then, kind of one of the key points is programmability. It has to be so that, I'm interested in gene A, maybe you're interested in gene B. How difficult is it for you to reprogram the system to recognize gene B, or gene C, or gene D, etc.? You want it to be super, super simple. And then finally, this is a kind of interesting point, is you want it to be easily accessible. So you don't want this technology to be tied up in lots of intellectual property rights, IP. You don't want that no one can use it. And I'll come back to that point a little bit in a moment. So there are a number of different enzyme systems that have been evolved and looked at to carry out this kind of function. And one of those is the meganuclease, which I won't talk about at all. So these are one of the first systems that people started looking at. But I won't talk about this at all. Well, I'll mention briefly, and you've already had introductions to these from Nigel Savory in the DNA recognition lectures. So zinc finger nucleases and talons. Two different types of programmable proteins that can be made to recognize specific genes. But the majority of these lectures I'll talk about tomorrow and next week is CRISPR. So the first idea in terms of making an editing tool people thought about is what we want is something that recognizes a specific sequence and then an element which does the cutting. So you want like a molecular scissors that cuts DNA and something which recognizes the gene that you can program. And so one of the kind of the nuclease part came from the restriction enzymes. You've already been shown this by Cara. This is FOC1. This is one of these type 2S restriction enzymes which recognize asymmetric sequences. So rather than being a palindrome, they recognize an asymmetric sequence and they cut downstream of this sequence here. And what they have is one domain in this purpley colour which recognizes the sequence, and this green domain, which is in this crystal structure, sort of piggybacking on the recognition domain, it swings out onto the DNA to cut here. So what you've got is a kind of separately folded domain which can act as a molecular scissors. So as biochemists and microbiologists realise if you just take that domain and attach it to other things, you can attach that activity, that nuclease activity, to that other protein, which could be a protein that you can program to recognise a different sequence. And what made this attractive, this particular type 2S systems, was the fact that this thing here is a kind of separate domain to the recognition domain. You've got one domain doing the recognition, another domain doing the cleavage. So what you therefore want to do is kind of say, okay, well, let's keep this DNA cleavage bit, which is from FOC1, that's your molecular scissors, and then make the grey bit something else which you can program to recognise gene A or gene B or gene C. And so the first idea, and again, I won't go into this in massive detail because we've had this already in Nigel Savory's lectures, is the use of zinc finger domains. So these are transcription factors which have this kind of folded motif where you have an alpha helix which recognises the DNA sequence through the major groove. It's coordinated by a zinc ion, and each of these motifs recognises 3 base pairs. So you have a motif, folded motif, and that recognises 3 base pairs. And so what was done was a library of these zinc fingers was made. This took a lot of effort, basically randomly mutagenizing and with sort of design, changing the sequence of the zinc fingers until eventually you got various different combinations of 3 base pair recognition. So then you have this library of these zinc finger elements that recognise AAA, ATA, GAC, etc. And you basically combine them together, you link them together to the nuclease. So here in this example there are 3 zinc fingers, blue, green and brown, which recognise a particular 3, 3 and 3 set of base pairs. And by linking together different elements within your library, you can therefore recognise different sequences. So if you want to recognise gene A, you say, what's the sequence of gene A? I pick this domain, this domain, this domain, link them together, and I can recognise that gene. And so then what you get is, in order to get double strand break, you have to have 2 of these FOC1 domains coming together, 2 of these molecular scissors, one cuts one strand, one cuts the other strand. And so therefore you have, in this little cartoon, 1, 2, 3 zinc fingers linked to one FOC1 nuclease domain here, and that dimerizes with another FOC1 nuclease domain linked to 1, 2, 3 zinc fingers here. So you have a specific recognition site, which is the red, purple, blue, brown, green, blue bit, which is 9 plus 9, so 18 base pairs. So 18 base pairs which you can program to be a specific sequence that you want. And once you get to 18 base pairs, you're starting to get a big enough recognition sequence that on average that occurs less than once per human genome. So you can start to get an enzyme which should, in theory, only bind at one location because you've programmed it to do that. And in this particular form, the zinc finger nucleases produce these overhangs, and this will feed into either the NAGJ or the HR pathway. So the problem with this technology, which kind of came around about in the 1990s, was that programming these things to work was very, very difficult. And in fact, what happens is a company, Sangoma, did a lot of the work towards making all these zinc fingers, and then they tied everything up with IP, so no one could use it. And that meant no one could test it and use it and evolve it and try and make it better. And so it's very, very difficult to get hold of these things and try and play with them. They could have held on to them and didn't let anyone use them. And they didn't have every single combination of 3 base pairs available. So it was a bit like there's certain things they couldn't touch, and it was imperfect. So it works. You can use it for gene editing, but it was imperfect. So the other system, again, which Nigel told you about, are the talons, which are based on these bacterial transcription factors secreted during plant infection. The tails, where again you have one of these repeated units, in this case helix turn helix, these tau repeats, and each of these tau repeats recognises now a single base. So this is a much simpler programmable system than the zinc fingers, because you can simply say, okay, I want to recognise an A, so you take one particular type of tau repeat. Now I want to recognise a T, you take a different one, and you link them together, and it forms these rather beautiful helical structures that wrap around the DNA. So you can recognise almost kind of very, very long sequences by linking these elements together. And exactly the same idea as zinc fingers. You build this thing with multiple repeats, and you then link that to the molecular scissors, the FOC1 nuclease domain. You have another one here to make a double strand break, so therefore you have 2 of these talons recognising sequences, and you can make very, very long recognition sequences with these talons. Here's a kind of one cartoon view of it, here's another cartoon view where these kind of beautiful helical structures wrapped around the DNA, linking together these nuclease domains at one precise location. So these are relatively easy to program, and there's not an IP tying these all together. The problem with this system is that in order to clone this and make the gene you want to produce the TALEN you want to cut gene A, you have to make repeated versions of these TAL repeats. Now, what are they made from? They're made from a gene which is made of DNA, and the problem is because the sequences are all the same, it's the same protein sequence repeated with some small changes where it recognises the DNA, but ultimately it's the same folded domain all over and over again. The DNA sequence becomes a repeat. It's a very, very similar DNA sequence repeated again and again and again and again to produce this repeated protein structure. And one of the things in cloning is if you're trying to clone DNA sequence that's repeated, it's really hard. Because you give that to a cell and the replication machinery starts making mistakes because you've got a repeated sequence. So repeated sequences are problematic. So this technology works really well. TALENs are a really good gene editing tool. They've been used in human patients successfully. So that people with white blood cancers have had their cells removed, gene edited using TALENs and put back in to try and target cancer cells, and this works. So TALENs work. The problem is they're not easy for everyone to use because of this problem of having to clone these repeats. It's tricky, which means the technology can be used, but you typically have to have an expert in the lab who's learned how to use technology. It's not something where you can just say, okay, knock that gene out using this system, and then we'll have the result next week. So TALENs is good, but it's not good enough. Which then leads us to CRISPR, which is then tomorrow. So hopefully, see you all tomorrow. We can learn all about how CRISPR works. Anyone got any questions, please do come and ask.